Introduction. Nucleus is the most important part of the cell situated in the cell cytoplasm. All the cellular activities are controlled by it. Nucleus is a directing and organizing using without which the cell could not exist. It was discovered by Robert Brown in 1831 in the plant cell. The nucleus and cytoplasm are interdependent and one can not survive without the other. The cytoplasm provides the most of energy for cell through oxidative phosphorylation and anaerobic glycolysis. The cytoplasm ribosomes contain most of the machinery for protein synthesis. The nucleus provides templates for specific synthesis mRNA and also supplies the other important RNA molecules like RNA and tRNA. First, the mechanism by which the genes contained in the chromosome exert their control on the metabolic process of cytoplasm, and the mechanism by which the cytoplasm influences gene activity. Experiment showing interdependence and interaction. Control of nucleus over cytoplasm. First, protozoa. It was first noticed by Balbini in Metomy experiment in which protozoa were enucleated and situated. Such enucleated fragments are able to sustain most cellular activities and they can form a cellulose membrane and carry photosynthesis, react to stimuli and ingest food. Active cilia extract. However, these enucleated cells generally survive only the short time within 5 to 10 minutes and are incapable of growth and reproduction. As observed in amoeba proteus, they are after removal of cell nucleus, its most has been slowed down, it can no longer digest food. When the nucleus transforms from an amoeba to another same species homotransfer activities of cytoplasm division, a mass culture may occur. Nucleo-cytoplasmic relationship in acetobularia. J. Hamberling in 1934 demonstrated that the nucleus controls the function of cells, ultimately character of individuals by conducting experiment on acetobularia. Acetobularia is a unicellular marine algae. Acetobularia has a stem between 3 to 5 cm long and a cap 1 cm in diameter. The nucleus is located in the basal or rhizoid end of the cell. In acetobularia, nucleus can be removed simply by cutting off the rhizome, and the resulting enucleated cells are active in the photosynthesis and can survive for many weeks. Each acetobularia species has particular gap morphology. If a nucleus from acetobularia created is implanted into an enucleated acetobularia Mediterranean from which the cap has also been removed, the resulting cell will eventually develop a cap of a acetobularia type intermediate. Here you can see the picture of acetobularia. Here you can Nucleo-cytoplasmic relationship in sea urchin. An unfertilized sea urchin egg enucleated by the being cut or centrifuge may be stimulated to divide without fertilization by sliding impression in hypertonic solution. In total absence of nucleus, it undergoes division to form a multicellular and a haploid embryonic form, which is regenerates at one which the nucleus forms it haploid and embryonic form. Upon division of cytoplasm done in the limited time can be continued presence of nucleus, but the nucleus is necessary for continued a normal functioning and the differentiation of cytoplasm of the cell. Nucleo-cytoplasmic relation in amoebian eggs. Amphibians eggs are most useful to study a mechanism of cell differentiation. During oogenesis, this is high rate of RNA synthesis, example tRNA, rRNA, mRNA, and it is says that during maturation, printed after cleavage and gastrulation. Pittsburgh and Gordon, 1968, transplanted the nucleus of differentiated intestinal cell tadpole of Xenopus leaves into a enucleated egg found in a normal adult frog, developed from the combination of the egg, cytoplasm and the transplanted nucleus. He converted upon the larval intestinal epithelial cell of the tadpole. Because of their large size, it is relatively easy to obtain undamaged nuclei from these cells. In Xenopus, the egg chromosome cannot be removed mechanically, instead they are destroyed by irradiation. Cytoplasmic control of gene expression. The nuclear transcription technique has also been used in examining the mechanism by which genes are activated, expressed during cell differentiation. Nuclei from blastula, gastrula, neural are given intestinal epithelium. The brain undergoes considerable changes when it implanted egg cytoplasm. Results depend upon the time at which nuclear implantation was. For example, if brain nuclei are injected in the global oocyte, the enlarged and rRNA synthesis is activated within them. If the transfer is made between the condensed chromosome, there is no change of implanted nucleus. If the transplanted nucleus is later failure during cleavage, the nucleus enlarges and starts to synthesize DNA. These experiments demonstrated in the implanted nucleus response according to the state of the site. Somatic self hybridization experiments show that the patterns of nucleic acid synthesis and gene expression by nucleus can be modified substance present in the cell cytoplasm. Red blood cells nuclei can be reactivated by the cell-fused and inactivated centivirus, a membrane, para-influenza virus, and others. The initial product of this fusion two different cells, heterokarya, a single cell containing a nuclei of two types. Eventually, both nuclei enter mitosis synchronously, form a single metaphase plate, divide and produce a hybrid cell line known as a synchron. The cells of hybrid cells line have single nucleus containing chromosomes from both parental nuclei. In 1965, S. Harris found that the chick erythrocyte nuclei are reactivated when fused to HeLa cell, an undifferentiated cell line derived from the uterine. Carcinoma cells chick erythrocytes are terminally differentiated cells have considered nuclei in the HeLa cell. Chick erythrocyte nucleus is increased in 20 times. Volume disappears in its chromatin. Resumes RNA synthesis, develop the nucleus and eventually replicate in DNA. So there is a conclusion. The above experiments clearly show that the synthesis of macromolecules in a nucleus controlled by the cytoplasmic environment. Only the cell cytoplasmic is required to reactivate the chick erythrocyte nucleus. Thank you for watching.